Hey class, like usual, a couple of announcements before we get started. First of all, your research projects are due tonight by 11:59 p.m. in Dropbox, and I apologize. I think in the last video I was saying they were due on the 16th, which is Thursday, but they're actually due the 14th, and so if you're just not realizing this and you're freaking out, send me a message. I'll help you out a little bit, but everything else had said that we were going to be due on the 14th, so we're going to stick with that unless that's a huge problem for you. Just let me know. We do have the test coming up on the animal unit on Thursday. That will be in mastering. Okay, so it's going to cover the whole animal unit that we started right just before spring break, and the review sheet is up on D2L, so you can access that and review for the test. As far as taking the test, it's not available until like 12 o'clock a.m. on the 16th, and you can take the test anytime that day. Okay, the time will be limited to, I believe it's 50 minutes. It will tell you when you log in there, it's 50 or 60. But you'll log in to mastering, you'll click on exam 3 and go in and just take that exam there, and you'll have, I think it's 50 minutes once you start it, okay? So don't go in, check it out, and then leave. Once you enter into that exam, the clock starts and you have 50 minutes to complete it. And you have to have it done before 11:59 p.m. Thursday night. Okay, so I don't care if you take it at 10 o'clock at night or 4 in the morning, that's fine. At any time you can take it, you just have 50 minutes to complete that. Okay, so I know this is, of course, like all your other tests on the honor system and all of that. So remember, keep your integrity, make sure that you're studying, because you do have to take some of this information with you to the next step, to the next level, whether it's university or whatever. So, and plus I don't think you're gonna have enough time to look up all the answers and things like that. If you have any specific questions about the test, let me know. Kind of the same format as if we were in class doing it. There will be a couple of true false. Mostly the test is going to be matching. There's a couple short answers as well. I believe there's 41 questions on there. And so that's kind of what the test is going to look like. Again, if you have any questions or concerns, let me know. If you have some serious test anxiety and you think you may need more than 50 minutes, which I really don't think you will, get in touch with me as well, but I think you guys should all be totally fine with that time limit. Okay, so today we're going to finish up our unit on the animal animal section, and we are going to finish it by talking about the immune system, or how the body defends itself from foreign invaders. Okay, so in anatomy, physiology, and microbiology, we cover the immune system as well. In micro, we have pretty much 3.5, almost 4 chapters on the immune system. And anatomy, physiology, we have a really big in-depth chapter as well. This is going to kind of just be gloss over basics. So those of you that have had other classes, this is probably going to be a good review for you. But let's talk about how the body protects itself against foreign invaders, okay? So for those that haven't had microbiology, well, we did probably talk about this a little bit when we talked about bacteria way back at the beginning of our organism unit, right after evolution. We talked about how the environment is just covered with microorganisms, right? With microbes. They're everywhere. We are covered in them. Our desks, everything, our phones, the air we breathe, everything is covered in microbes. And they're bacteria, they're protists, they're fungi, and viruses. And remember, viruses are non-living organisms. And most are going to be, if you remember, harmless. Most microbes we encounter don't hurt us. We just basically ignore them and they ignore us, and it's no big deal. But some microbes are pathogens. And here's one definition that is on your review sheet. And a pathogen is basically a disease-causing organism, or you could say microorganism. Okay, so pathogens, once they enter your body, will start to, excuse me, reproduce, metabolize your cells or parts of your body for nutrients and energy. And then once they've reproduced and established an infection in there, in you, then they'll want to spread on to a new host. And so that's kind of the cycle there. Your book talks a little bit about invertebrate animals and how they defend themselves, and we're going to just skip that part and specifically talk about humans. But we will kind of see, since humans are vertebrates, some crosses into other vertebrate animals as well. But vertebrate animals, including humans, have three major lines of defense. Okay, 3 lines of defense. And these are something that you'll need to know for your test as well. The first line of defense is going to be external barriers. Okay, I always like to think of the host's body as like a fort. Okay, and if you think of, you know, like in medieval times, they had a tall castle wall, they had a moat, they had a drawbridge, they had lots of barriers to prevent entry. And so that's the first line of defense, like keeping things out. Okay, so be barriers like the skin and also cilia, which are like little hair-like structures on cells. Like in the trachea, if you breathe in dirt, the little cilia will flip the dirt up and you can cough or spit it out. Some other barriers are secretions like tears. Tears would be kind of like the moat, right? It washes the things away, washes foreign things away. Same with saliva and mucus. Mucus traps dirt and foreign bacteria, pathogens, all that kind of stuff that shouldn't be there. So you can put it in one place and you can expel it out of the body. Okay, so the barriers is the first one. The second one, barriers. The second line of defense includes cells and processes. And then the third line is what we call the adaptive immune response, which is going to be the T and B cells. Okay, and B cells produce antibodies, which we'll talk about those in a little bit. Now in micro and in our anatomy class, innate immunity includes both the first and second lines of defense. And I prefer to think of it that way because innate immunity is something that you're born with. Okay, so unless you have some major congenital birth defects, you know, you're not going to have areas of skin missing, right? You're born with skin because you're a human. You're going to have things like inflammation and fever, and so some of those things that are going to be present at birth, basically. Oh, excuse me, I get to talking in the afternoon and I start yawning like crazy. So the innate immune system is something that you're born with, and it has general defenses, meaning that if you want to put in parentheses somewhere, if it detects something foreign, it just attacks it and keeps it out. It doesn't say, oh, you are influenza type A with this particular S and, or I'm sorry, H or N protein. Okay, it's like, oh, you're foreign, you don't belong here, you stay out. Okay, whereas the adaptive immunity, which is the third line, is acquired with exposure. So you're not born with it. It's something that you build up over time, and it's very specific. and it has memory. No memory is made with the first and second lines of defense. So the cool thing about the adaptive immune response, or the third line of defense, is that it will remember that pathogen and it will remember how to fight it, and it will be so good at fighting that pathogen that when you're exposed to it again, you may not get the disease again, or very, very mild symptoms. And so that's where we get the effectiveness of vaccines, is because our bodies create a memory. So 3 lines of defense, make sure you know which ones or what they are: the barriers, the cells and processes, and also the adaptive immune response. So basically what happens is, say you get a cut and the bacteria can breach your barriers, your first line of defense, breaches the skin. It comes in. Your area, your tissue might become inflamed. You'll have some phagocytes coming over to try to help take care of that infection. And if it's still not working properly and doesn't get it cleaned up in the matter of hours or minutes, then the adaptive immune response will come in. This is kind of like the special ops team, okay, that comes in and kind of does the cleanup. They get all the intelligence, there's memory made, all that kind of thing. So let's look at these a little bit more specifically. Okay, so non-specific, okay? It's just anything that's foreign, it attacks. Okay, so if the external barriers are breached, the innate immune response, and I would just call that the second line, because I would personally consider the first line of defense and the second line of defense innate, but this is what our book has. So if the first line gets breached, like we saw, the second line comes in, okay? So some white blood cells, as part of the second line of destroy are going to engulf foreign particles, or also destroy infected cells, okay? Now, a fancy term for white blood cells is leukocyte. Okay, so we have red blood cells, which are called erythrocytes, that carry oxygen throughout the body. Okay, white blood cells' job is to defend the body. Okay, there's many types of white blood cells, many types of leukocytes. Some of them engulf, and those are called phagocytes. And others destroy infected cells, like what we call NK, or natural killer cells. Okay. Another thing that's going to happen with the second line of defense is chemicals will be released by those damaged cells. Okay, so if you have an injury or a pathogen attack where your cells are being damaged, they're going to release some chemicals, basically messages to the rest of the body saying, help, I'm under attack. And these are called cytokines. And there's many types of cytokines. There's an E. And we're not going to get into like what their names are and all the different ones, but there's many different kinds of chemical messages that damaged cells will release. And they're going to tell other white blood cells or other leukocytes, hey, come to this site of infection. Hey, let's get inflamed and let's have a fever. And those are both ways to help protect the body. Specific internal defenses are those that are a part of the adaptive immunity. Okay, and that's basically like our last resort, our third line of defense. Okay, so they will selectively, very specifically identify what pathogen it is, or it could be a toxin as well, like tetanus toxin. And then they will also remember this invader. Okay, and then it will have a faster response time in the future, where we get the premise for vaccines. Oops, can't even spell. And then whether you're getting this immunity from a vaccine or an illness, you get that faster response time. And that's going to cause you to have no disease or more mild symptoms the second or subsequent time that you are exposed to that same exact pathogen. Now, we're going to talk about vaccines here in a little bit, but the reason that we have to get a flu shot every year, the reason that we don't remember the flu from year to year to year, influenza from year to year to year, is because it mutates and it changes. And so it tricks our immune system. And what the immune system, say you got the flu last year, the immune system remembers that flu, but the new flu has mutated so much that the body doesn't recognize it anymore. And the body is so specific in the way that it remembers that it doesn't identify the new flu unless you've been exposed to it years and years ago or through a vaccine or whatever. Okay, so one thing that I'd like you to remember is the differences between the different lines of defense, differences between innate immune response and adaptive. Okay, so here's a chart that's going to kind of help with that. Is the innate immune response specific? Nope, not going to be specific when you are considering an invasion. The adaptive immune response is speed of response. This is another big difference. Innate immunity is almost immediate, happens very quickly. This takes 1 to 2 weeks to get in gear. Okay, so with the initial first invasion, the very first time you're exposed to something, it takes a while to get the maximum effect of this adaptive immune response. But by the second invasion, this is going to be the same because there's no memory. Okay, but this is going to be really fast. This is going to be down to a couple of days, maybe at the most. And so the body has a time to react very quickly. Okay, and hopefully you won't have any disease symptoms with that, or at least it will be less severe than it would be. Okay. Types of white blood cells, types of leukocytes. LEU. I wish I could write better on here. Okay, there's several different types. I'm not going to make you know all of them. Most of these are going to be involved with the innate immune system. Let's see. For the most part, the B cells and the T cells, which we'll get into in the next slide, are going to be part of the third line of defense. And for the most part, these other ones are going to be part of the innate immunity or the second line of defense. Most of these are phagocytes. Anytime you see they engulf here, they're going to be phagocytes. NK cells, we mentioned those, they destroy infected cells. Mast cells, if you've had allergies, those are your enemy. They're going to release histamine. That's going to cause the allergic reactions that you have, the itchy eyes or whatever. Okay. B cells, there's a couple of types of these cells. They come in different versions when we look at the third line of defense. B cells, or sometimes called B lymphocytes. Okay, so we have leukocytes, which is the big category, and B lymphocytes are a type of white blood cell. There's the memory B cells that stick around and try to remember and fight off the infection really fast next time. And then there's plasma B cells that are going to make antibodies. And I'm sure you've heard about antibodies before, and they're going to be really important in keeping the body safe. And we'll talk a little bit about those as we get to them. The other type of leukocyte that's involved in the third line of defense is the T cell, or the T lymphocyte. Okay, there's four different major types of T cells. There's the cytotoxic T cells, or sometimes those are called the killer T cells. And they're going to destroy infected body or cancerous cells. We'll talk about how they do that. The helper T cells have to stimulate or activate the B and the killer T cells. We have memory cells, and then we have regulatory T cells that kind of tone down the response. Okay, keep the immune system from attacking yourself, which is bad. That's called autoimmunity. It causes an autoimmune disease, so we don't want to attack ourselves. And it also says, okay, the infection's over. Let's everybody chill out. We're done. Stop being on the defensive. Okay, or the offensive, either way. So we have the memory cells that help stay and remember for next time. The helper T cells are very important. Both the cytotoxic T cells and the B cells must be activated before they can act. B, and that stands for cytotoxic T cells, must be activated by T helper cells. Okay, so people that have HIV infection, human immunodeficiency virus, that virus attacks the helper T cells. Okay, what kills the HIV patient when they experience full-blown AIDS is usually some very, very mild infections that other people have with healthy immune systems that they may not even show any symptoms for, because their immune system is competent and they can take care of the person. The HIV attacks and destroys the helper T cells. Once the helper T cells are destroyed, and there's not very many of them, the cytotoxic T cells and the B cells that make the antibodies can't fight for the body anymore, and the body succumbs to usually a very opportunistic infection. Okay, so as we said before, the innate immune response responds non-specifically. Okay, and once the pathogen has penetrated those external barriers, the skin, the mucous membranes, mucus itself, and so forth, it will encounter three different things. Okay, so this is the second line of defense. Oops, go back. It will encounter the inflammatory response. It will encounter the fever response, and also white blood cells. Okay, those ones that we said were the second line of defense. Okay, so these are three things that are going to attack that invading pathogen. Okay, so we said most of those are phagocytes. Phagocytes and NK cells are going to destroy those microbes. And again, like we said, a lot of those are called leukocytes. Okay, so of the leukocytes, the phagocytes are going to ingest through phagocytosis. And I believe we've talked about phagocytosis a couple of times. That's where, and I'm terrible at drawing, but say we have a bacterium here. A phagocyte is a cell that's going to, basically through a process called chemotaxis, move toward an invader, and it will send out little pseudopods to surround this little bacterium here, and it will bring it in to its body, Okay, it's also going to fuse another sac or a lysosome with this little sac that has the bacteria trapped in it, and this has digestive enzymes and sometimes hydrogen peroxide, acids, and different things like that that will digest and destroy the bacteria or whatever the foreign invader is. Okay, so that's the process of phagocytosis, is they go around and they just engulf anything that's foreign or anything that needs to be recycled. And some examples of phagocytes, we've seen those in the other chart, but macrophages, neutrophils, and dendritic cells are the three major types of phagocytes. And here, these aren't my favorite pictures, but here's a bunch of bacteria inside of a phagocyte that has been ingesting them. Okay, so that's the job of the phagocytes. An NK cell is another type of second line of defense white blood cell or leukocyte that targets harmful viruses. Remember, viruses have to live inside of the host cell, right? They can't do anything without being inside of that host cell. And so once they get in the host cell, they take the host cell over and basically use it as a virus manufacturing center. Do you guys remember that? Okay. NK cells will detect and destroy virus-infected host cells. Okay. So they often are able to destroy the viruses before they've had enough time to reproduce and burst open that cell. Remember, I believe we talked about how a virus here's a host cell, the virus will come in, take over, and we can maybe sometimes have a couple of hundred viruses in here before that cell will burst open. Okay. Once the virus has hijacked a human cell or a cat cell or whatever the host cell is, that virus causes that cell to just be a virus manufacturing center. So that cell is not useful to the host anyway. So the NK cells come over here and they basically destroy that infected cell before they can reproduce and burst out 200 more viruses or virions. Okay. Same thing with cancer cells. NK cells and also killer T cells can identify cancer cells sometimes and destroy those as well, or any other type of abnormal body cell. So how do they do that? How do they detect whether it's a nice healthy cell or a cancerous cell or a cell infected by a virus? Well, remember, all cells have little receptors on them that identify the species, the individual, the type of tissue, whether it's a muscle tissue or mucous membrane or whatever. Okay, all of those surface receptors are giving the immune system, giving pathogens, giving other host cells an identifier of what type of cell that is. So normal body cells display a set of proteins, like little receptors on the end, called major histocompatibility complex, or MHC proteins. And these are basically proteins that allow the body's immune system or the body's cells to identify them as self. Okay, so if you have to have an organ transplant and you don't have an identical twin, what they will try to do then is look and see if they can find a match for you. A lot of times it'll be a close family member, like a brother or sister. But what they're doing is looking for MHC proteins that match. Because if I were to give you my kidney, which I would do if you needed a kidney, you could have my kidney. But we're probably not a match, unfortunately, because my MHC proteins are probably going to be different than yours. I would still give you my kidney though. And so that's why sometimes being on a transplant list takes a long time to find a correct match, because you have to have those, the organ that you're going to be receiving has to match your other cells. Otherwise, the immune system's like, oh my gosh, this new kidney is a foreign object and we're going to destroy it. Okay, so you have those MHC proteins to help the immune system identify what's normal and healthy and what's not. And so a lot of times viral infected cells lack some of the right MHC proteins. So they look different to the NK cells, okay? Because they're producing viruses and, you know, they may have lost some of their MHC proteins due to just natural degradation. And so the NK cells are like, whoa, this cell's not looking so good. Maybe we should destroy it before it releases a bunch of viruses. Cancerous cells will either have missing or altered MHC proteins as well. So sometimes they're identified and destroyed. Okay. Now, besides those cells, there's some other chemical responses that can take place too. One of the cytokines, and your book doesn't, well, I don't have it printed out in our slides, but one of the cytokines or one of the chemical messages is called interferon. And if you've ever had influenza or any other type of viral infection, you may feel really run down and achy and just really lethargic, and you just have a headache and you feel miserable. What's causing that is called interferon, and it's a type of cytokine. It's a chemical message that cells send out to each other and says, hey, I'm infected by a virus, shut down your metabolism, because if this virus gets into you, you are going to start making baby viruses. And so that signal, the interferon signal, basically shuts down a bunch of cells to prevent the virus from hijacking them. So I thought that was kind of an interesting note as part of the second line of defense. Another important part of the second line of defense is inflammation. Okay, inflammation or inflammatory response attracts phagocytes to injured or affected tissue. So if you injure your finger, for example, okay, it's going to get inflamed a little bit probably. And what that inflammation does is brings more phagocytes, brings more white blood cells to that area to help with repair, and also to help with limiting the spread of infection. Okay. So let's see, some major indicators of inflammation. Okay, the site will be warm. Okay, so think if you've ever had an infection, you've probably cut your finger before, and you know, think of it. What happened? It became warm and red, probably swollen too, right? And maybe a little painful because the pressure from the leaky blood vessels and all the swelling puts pressure on those nerve endings and makes it a little bit more tender than normal. Okay, so we mentioned mast cells. They help with allergies, but they also stimulate inflammation. And they're going to release histamine, which is another chemical mediator, another cytokine, and the histamine causes the inflammatory response, the inflammation to happen. Okay, so here you get a splinter. Remember, bacteria is on everything. Gets some bacteria deposited here. The skin is damaged, so those damaged cells are releasing chemical messages. Here's our mast cells that just happen to be in the area. They're going to start releasing histamine. Histamine is going to cause this little capillary, this little blood vessel here, to become leakier. You see these little guys sneaking out here? This vessel becomes leakier. Also, you get fluid leaking out here. That's where the swelling or the edema comes from, is this leaking out. And that leakage helps these phagocytes and other white blood cells get out of the bloodstream and into the site of infection. Okay, that process, for those of you who are super into science, is called diapedesis. And that's basically just the movement of macrophages and other phagocytes out of the blood vessels, squeezing out to get into the site of infection. Okay. Fever is another type of secondary immune response, and this is going to be, you know, if you cut your finger, you're probably not going to get a fever, right? Unless you get some sort of large-scale systemic major infection. Okay, so fever is kind of one of those that you don't always get if it's just something minor. Okay, it's usually something a little bit more widespread when you see a fever develop. Okay, so these foreign invaders can be made up of what are called pyrogens, and these are chemical messages like a flagella or protein or a toxin or something that triggers a fever, which both a fever is very helpful in some circumstances to slow microbial reproduction and also slows and enhances the body's defense systems. Okay, let me see. Yeah, we'll talk about that here in just a second. So fever, well, back up. Remember homeostasis from the last chapter we talked about? Maintaining internal environmental conditions, okay? The hypothalamus maintains a lot of internal conditions, and temperature of the body is one of them. And it is a portion of the brain that basically detects temperature and sets the thermostat. Okay, in humans, thermostat, they say is 98.6 degrees Fahrenheit, but there's actually some evidence that it's actually a little bit lower in modern humans, which I guess we won't get into that now for the sake of time, but somewhere between 97, 98 degrees, 99 is normal human body temperature, 37 degrees Celsius. Okay, so some types of bacteria and other pathogens have chemicals called pyrogens, and basically means fire, fire starter. Pyrogens travel around the blood, and the hypothalamus detects those, and it resets the body's thermostat set point. Okay, so instead of, say, 98 degrees, the hypothalamus says, oh, we have got some sort of toxin here. We need to combat this and fight it. And so it will maybe reset that thermostat to 101 degrees Fahrenheit. And then think about when you've had a fever before. What has happened? You get the chills. It may be 90 degrees in the bedroom or wherever you're hanging out feeling terrible, and you've got 13 blankets on, a sweatshirt, you know, your hood pulled up and all that, and you're still shaking, right? That's the body's way of raising the body temperature. And then once your fever breaks, what happens? Hot. Take the blankets off. You sweat like crazy. All of those types of things. Those are the ways of the body basically just bringing up the temperature and then bringing it back down. Okay, so think about enzymes when we think about fevers. Okay, so it's said that a fever will slow microbial growth. Well, if you get too hot above the optimal temperature, right, it will slow down pretty darn quickly. But also, so will our enzymes. Okay, so remember this graph when we talked about enzyme function. Okay, we get to the optimal temperature and we drop off pretty quickly. Okay, we can have a slowed enzymatic response here. They can work okay, but here they get denatured. Okay, so with fever, we have to have this kind of this sweet spot where we have slow microbial growth, but we don't destroy our own enzymes. So if you get over 104 or greater, and you have that for a prolonged period of time, that can be very dangerous to the body. 109 degrees or more is fatal to the human. So a high chronic fever, let's write that, high or chronic fever is detrimental, sorry. Okay, a lower fever, 99 to maybe 101, might actually be helpful and might not be something that you want to take a Tylenol for to try to reduce that fever, because the fever is actually helpful. Same thing with inflammation. What we call acute inflammation. Okay, so short-lived inflammation, say you sprain your ankle, a little bit of inflammation is probably good because it's going to bring some chemicals and things to the site to promote healing. But if a month from now, if your ankle's still swollen, that's probably not going to be very good for you. Chronic inflammation is actually damaging to tissues. So acute, fast, mild inflammation or fever can be helpful, but lots of inflammation, high fever, long periods of time is actually damaging. Okay, so that's the innate immune system, the first two lines of defense. We didn't talk a lot about the first line, the barriers, but that's basically the skin and the mucous membranes. The third line of defense, or the adaptive immune response, is going to be used several types of cells, or several types of white blood cells. Okay, the main ones we're going to focus on are the lymphocytes, the T and the B cells. Okay, they're the major players. Macrophages and dendritic cells, these are going to be signaling cells. Okay, they're going to basically signal the helper T cells and the B cells, like, hey, we've got a foreign invader over here, you should come check them out. Okay, so they play a role in both systems, innate and adaptive. Okay, the lymphocytes are only a part of the adaptive immune response. And again, it's the T and the B cells. They're made in the bone marrow. And they hang out a lot of times in the lymphatic system. Okay, the lymphatic system is kind of similar to the circulatory system in that it's made up of a whole bunch of vessels, and its job is to return fluid back into circulation. So you do sprain your ankle, okay? One way to get rid of that excess swelling is to send it back up through the lymphatic system and a little bit through the cardiovascular system, and return it back into circulation. Another part of the lymphatic system is lymph nodes. And lymph nodes basically filter all of that fluid that's coming through the lymphatic system and do surveillance. It's kind of like a checkpoint, the TSA checkpoint going through the airport. Okay, they're checking for foreign things. If there's something foreign there or it looks abnormal, the immune system will attack it in the lymph nodes. So if you've ever had like an upper respiratory thing and you go to the doctor and they feel your tonsils here, they feel your lymph nodes, if they're enlarged, you're probably fighting an infection because they increase in size during that time because there's a lot of white blood cells and other things there trying to help with that infection. Let's see. So there are many different proteins. We see proteins a lot involved in the adaptive immune response. There are many different types of cytokines, which are proteins. Remember, those are the chemical messages that are produced by a large variety of cells that provide communication. Okay, and there are some that are used to stimulate the T and the B cells. Okay, so types of proteins, cytokines. Another type of protein is an antibody. Okay, so antibodies, as we remember, are produced by B cells, and they're going to help recognize microbes and destroy them. And we'll see how that happens here in just a little bit. But those are some of the important proteins when we look at the adaptive immune system. Okay, so all adaptive immune responses, whether it's the T or the B cells, are going to have 3 steps, and you'll need to know these. It is on your review sheet as well. So the first thing that happens, whether it's a T cell or a B cell, is the T or B cell, the lymphocyte, needs to recognize the foreign invader as non-self. Okay, then they launch the attack, and then also they retain a memory of that invader for next time. Okay, so those are the 3 main steps: recognize, attack, remember. So step 1, recognition. Okay, the immune system, as the adaptive part, recognizes the invaders, basically proteins, well, sometimes complex carbohydrates, but a lot of times proteins. And there are large molecules that are usually complex, they're really easy to identify, that trigger the immune response, and these are called antigens. Okay, so that's the definition there. Any molecule that can trigger an immune response is called an antigen. Okay, so an antigen would be like, you know, if an enemy is approaching you and they have a gun, the gun would be the antigen. Okay, if another enemy is approaching and they have, you know, a knife, whatever, that's something that you would recognize as a threat, right? Okay, so an antigen is something that the body recognizes as foreign and basically worthy of attack. Now, remember the dendritic cells and the macrophages? Okay, they are involved in this recognition part, and they're going to display antigens from those microbes that they've engulfed. So they're going around, they're phagocytes. They're going around engulfing bacteria. Okay, as they do that, they're going to take that knife or whatever the antigen is and display it on their cell membranes. So here's a dendritic cell. They've kind of got like little fingers here. It's phagocytized this bacteria here, and it has little plus signs as an antigen. What it's going to do is it's going to display that plus sign out here. Okay, that's called antigen presentation. Other antigens like toxins, okay, might be released by the bacteria and float around in the body. Okay, and any large molecule, okay, including molecules of your own body cells, may be antigens as well and determine and stimulate an immune response. Okay, a lot of times we don't want that to happen, but it does. Okay, once antibodies and T cells are triggered, those lymphocytes will produce 2 different types of proteins that can bind to a specific antigen and trigger an immune response. Those proteins are the antibodies made by B cells and T cell receptors. Okay, so these are basically receptors on the cytotoxic T cells. Okay. Here is what an antibody looks like. It's made up of different chains of proteins and sulfur and all this different stuff, and I'm not going to make you remember it, but an antibody basically looks like a Y. And then on the end, it's going to have little antigen binding places. Okay, so on antibodies, you can have 2 antigens binding. You can have one here, which is this yellow area, and you can have another one that binds here. Now, because the adaptive immune system is very specific, these antigens, okay, have to be the same to similar, or two antigens of the same type, I should say. A T cell receptor is found on the surface of a T cell, a cytotoxic T cell, and it only has one binding site. Antibodies have two, T cells just have one. Okay, so remember that, again, adaptive immune system is super, super specific. And just like the protein's enzymes, right? They're super, super specific because of their active site. So they won't bind to something that's not the correct shape. Same thing with T cell receptors and antibodies. So each binding site has a specific shape, electrical charge, specific size, and will only allow a certain antigen to fit. Okay? So they have to bind to only one type of antigen. So every B cell produces its own unique antibody. Okay? Same thing with the T cell. Now with antibodies, there's 5 major classes. There's IgA, and Ig stands for, let me write it over here, immunoglobulin. IgA is going to be an antibody found in secretions like breast milk. There's IgD, not really quite understood what those antibodies do yet. There's IgE, and those are going to be involved with parasitic worm infections and allergies, is where you see high levels of IgE antibodies. There's IgG. This is the longest lasting antibody. It's also the most numerous. And it is one that can cross the placenta and help baby develop some protection. Okay, and then the last one is IgM. IgM is the first responder. It's usually the first one on the scene. And it is the largest in size. Okay, it's usually the shortest lasting of the antibodies, but those are the 5 types of antibodies. And so what do antibodies do? Okay, we said that they recognize foreign invaders and trigger a response against them. Okay, so they can bind to foreign things, and they can trigger other immune responses like complement. If you had one of the other classes, you might have heard of that. The complement cascade can also trigger phagocytes to come over and help as well. And they can help destroy those invaders as well. Let's see. I'm just going to skip that one for now. T cells, though, have a T cell receptor, the other type of protein. And T cell receptors are going to be different or differ from all other T cells. Now, that's kind of a confusing statement because every T cell, if you go back up here, B cells get activated when helper T cells activate them, and also a microbe would bind here, and then there's going to start to produce antibodies. Okay, this is what we call a B cell BCR, a B cell receptor. Okay, that's not an antibody. This B cell will only have one particular type of antibody that's released. Okay, T cells are kind of the same way. They only have one type of receptor. Okay, so this one will only bind to oval type antigens. Okay, the next cell over here may have one that attaches to triangles or whatever, okay? Again, it's very, very specific, but each cell has a specific type, if that kind of makes sense. Let's see. So the immune system is really kind of amazing because you have millions of different T and B cells, and so you can potentially respond to millions of different potentially harmful antigens. So I have T and B cell in my body, and so do you, that will help combat jellyfish stings, okay? I've been to the ocean a couple of times for just a few brief moments. I'm definitely not a water person, okay? Didn't grow up around the ocean. My family's always been landlubbers, you know. But I do have T and B cells that will recognize jellyfish toxin, okay? Now, if I get stung by one, my T and B cells will recognize it, but they're gonna work too slowly to be able to help me. Okay, if that kind of makes sense. But you have millions of them that identify millions of different types of antigens. And at any given time, there may be 100 million different types of antibodies and even more T cells that identify different types of foreign things. Okay, so how does your immune system recognize those invaders? Let's see. Why don't they attack your own self? Okay, that's where we are. Sorry. Again, we talked about those MHC proteins. Okay, but one thing is too that you have a continuous presence of your own antigens during immune cell development. Okay, there's a lot of research right now going into what we call the microbiome and how the microbes living in and on our bodies affect our immune system development and all kinds of other things as well. We talked a little bit about that last time about the gut microbes, okay? So our cells have been there all the time because remember, this is an adaptive process. It develops over time. So they're not new, okay? Another thing is there's a regulatory process done by what are called regulatory T cells. And I'm not going to go through the process, but basically what happens is it is a check system and self-reactive T or B cells are killed through apoptosis, or basically programmed cell death. Okay, so there is a check system, and it doesn't always work because sometimes people develop autoimmune disorders, but there is a system that try to check and make sure that the body doesn't attack itself. Okay, so how do they attack invaders? So they know how to not attack yourself. How do they attack invaders? Well, before we get to that, we have what's called the humoral immune response, and that's going to be the B cells and the antibodies. And then we have what's called the cell-mediated immune response, and that's going to be the T cells. Okay, so we're going to take a second and look at the humoral immune response first. Okay, and basically what happens is with the B cells, and they haven't shown here how the helper T cells activate them. Activate. But they've kind of left that process off, just for simplicity. But say you have an antigen here. Say this is a bacterial toxin, okay? These here are, they call them antibodies, but really they're B cell receptors, okay? They're going to bind with any of these cells that have the correct shape, okay? Now, this guy here is going to say, oh, look, I've recognized these Now I need to multiply. This is basically called clonal expansion. They make little clones of each other because we need more of this one. We don't need more of this purple or black. Okay, so they'll start cloning themselves, and some of them develop into plasma B cells, which produce the antibodies, and these guys are memory cells, so that they can stimulate the reaction a lot faster if you're ever exposed to that particular antigen again. Okay, so differentiation into memory B cells and plasma B cells. Those are the ones that are going to make lots of antibodies, like 2,000 antibodies per second. They make a ton, and then those move around in the bloodstream and go to wherever the infection needs to be. Okay, so humoral immune system is the antibodies. Okay, the B cells that produce the antibodies. Again, how do the antibodies work? This is a toxin, okay, a snake bite toxin. Okay, the antibodies can block here. Here's the active site in that enzyme to cause damage to your neurons. It's a neurotoxin. So if the antibody blocks that, it basically can't bind. Okay, it's called neutralization. Another one is that they can coat that microbe and it flags it a little bit more for the phagocytes to eat them up. They can cause the complement cascade for those that have had the other classes. Okay. So humoral immunity, the antibodies, which are made by the B cells, are going to fight invaders that are outside of the cells. Okay, that's really important. Okay, they do not enter cells. They fight on the outside of the cell. Okay, so they wouldn't go into any of the buildings, okay, or any of the rooms inside of the fort. They stay outside. They're in the moat or inside the fort walls, but they're not going to go into any of the buildings, okay? And these are going to look for microbes themselves, or they're toxins that are just kind of floating around wherever, okay? But they cannot go inside of the cell. The cell-mediated immunity, or the T cells, can, though, go inside of the cell. And remember, they attack virus-infected body cells or cancer cells. Okay, and their job, what they do at the end of the day, is they're going to lyse the cancerous cell or the viral infected cell. Okay, they have, let me see, do we talk about that? It's called perforin granzyme. They basically poke a hole in the cell membrane of the infected cell and burst it open, lyse it. Okay, let's see. Helper T cells help with this whole process. You can cross this page off for the interest of time. Here, I'm going to let you guys look through this. This is how both of these processes work. We kind of looked at B cells. T cells are the same way. They get stimulated by helper T cells. It's going to clone and then turn some of the clones into memory cells. Some are going to be into cytotoxic or killer T cells. And then here is the hole that it's punching into this infected cell, and it's going to lyse that cell. Okay, so here's a T cell and a cancer cell. So even after you've recovered from a disease, perhaps for years or maybe your whole lifetime, you can remain immune to that disease. Now, not always, but for a lot of things you can, and that's thanks to the memory B and T cells can survive for years. if the body is re-invaded. Okay, the immune system is going to be faster response, and you may not get sick at that time. Okay, so the first exposure this is levels of antibodies, or what we call titers. First response takes a couple weeks to peak, and then look, the peak's not real high either. But if you get a booster shot, or maybe you've gotten exposed to it again, okay, here's your second exposure, time 0. Within first few days, look how strong that response is, and look how quick it is compared to before. Okay, so make sure you look this over. This is basically the premise of why we vaccinate. Okay, so a vaccine stimulates an immune response, basically artificially just gives you that, antigen so you can remember it. Okay, so vaccines are going to consist of weakened or killed microbes so that they don't make you sick, but you're getting the antigen to mount the memory. Okay, so memory is produced through the T and the B memory cells, and then again, I feel like a broken record, produces a rapid response. Okay, I think we're gonna skip allergens and cancer. And I think we will just stop from there, okay? Because this lecture is taking forever. I'm going to double check the review sheet and make sure there's nothing on there that comes in talking about any of the allergies or cancerous stuff. But I think I've overwhelmed you with too much. So we're going to stop there. I will double check the review sheet. If you've already printed it off and looked at it, you might check back to see if it's been updated. There'll be things taken off, not put it back on. So if you have any questions about the review sheet, if you have any questions about the test or about the research paper, just let me know and I will try to address those for you. Otherwise, log in on Thursday. You'll have 50 minutes to take the test and we'll go from there. We're going to talk about the ecology stuff next week. All right, so have a good day and we will talk to you soon.